and welcome to your lecture video for Chapter 12, the Blood Chapter. This is the first of two chapters on the cardiovascular system. Chapter 13 will cover the heart and blood vessels. So just to get started, blood is a type of connective tissue. If you remember, it's the fluid connective tissue. That matrix that everything exists in is your plasma, and then the formed elements we're going to see are things like your red blood cells and white blood cells. So the cardiovascular system is comprised of both blood, heart, and blood vessels, making your circulatory system. We kind of consider it the same thing: cardiovascular, cardio heart, vascular vessels, and the blood is pumping through all of that. Your blood is what's transporting substances. So it is that, it's not proper connective tissue, but it really is connecting different places. It's transporting stuff from one place to the next, circulating it through your organs, making sure things have nutrients, any waste are being carried away. So it provides this crucial function for us, and our body works hard to make sure that our blood volume and formed elements within the blood stay at a proper level. The gas main cells, sorry, not gas transporting, yes, the red blood cells, but the other cells, white blood cells for fighting infection, platelets for clotting and stopping bleeding, and then as we said, the plasma, everything exists within that. Within it as well are the nutrients, that things like proteins, vitamins, minerals, any other kind of hormones that might be transporting through the body. Those things are also being moved in conjunction with metabolic wastes. All of those things, the red blood cells, white blood cells, and platelets, are your formed elements. They get produced in the red bone marrow of the long bones in the skeleton in an adult, and we'll see in other places for the fetus. Here's just an overview. We'll see this image again in a couple of slides about the breakdown of blood. So within your blood, you have these formed elements and then your plasma. Plasma makes up about 55% of total blood volume. Formed elements is 45%. And of this 45%, it's red blood cells that make up, as you can see, the vast majority of it. So most of the formed elements, that's why your blood tends to reflect the color of your red blood cells. It's because they are so high percentage representation. White blood cells, 0.1%. This fluctuates somewhat depending on what kind of, if you have an infection or something like that, then it might be affected, but really it should not be too high. And then platelets are about 4.8%, 5%, and they're again for clotting. Of the white blood cells, we're going to learn the 5 different types. Each one, you can see, has their own concentration. It's not an equal representation, depending on, again, what kind of infection or lack thereof, hopefully, that you have. These numbers might change. Within plasma, you can see some of the breakdowns with that: electrolytes, water, proteins, waste, nutrients, and gases, and then each of those broken down. So we're going to look at all that. So looking at blood volume and composition. So as we said, plasma, that fluid portion of blood, is mostly water, but then you have all of these other things that are floating and dissolved within that or being transported through that. Blood pH is about 7.35 to 7.45. So is that more acidic or alkaline? It's more alkaline, yes. So it is more basic than standard neutral water, which is 7. This is when I get to take just a moment to talk about pH-adjusted water. I never really understand what that's supposed to be adjusted to, because standard water is 7 as a neutral pH. And so is it pH balanced to 7 already, or which is actually acidic for your blood? Or is it more alkaline? And if so, how much more alkaline? And also, when you drink water, it goes into your stomach, and your stomach has a pH of about 2 or 3. So anything that you drink is just going to get reconstituted into the normal pH of whatever liquid it's within. So electrolyte enhanced water, smart water, yes, that's great. But pH balanced water, what is it really doing for you? And your body should not have any problems maintaining its own pH. If your pH, so normal is 7.35, if it gets down to about 6.8, so just a 0.5 change in the pH level, your proteins start to break down and you start having organ failure. Kind of same thing with the upper limit as well. This is a very, very tightly controlled range, and so any kind of major shifts you will definitely start to notice, and pH balanced water will not help you. All right, so the percentage of formed elements in the blood is normally about 45%, like we were saying, between your red blood cells, white blood cells, and platelets. It's about 45%. That's called your hematocrit. Okay, so your HCT. So you might hear people talk about the hematocrit within the blood, and that's just talking about mainly the red blood cells. And I believe it's clinical hematocrit is specifically referring to the red blood cells. If you just say hematocrit, it's technically supposed to mean red, white, and platelets, but most of that is going to be your red blood cells. White blood cells and platelets make up less than 1% of total blood volume when you combine it with the plasma as well. And an average size adult will have about 5 liters of blood. That can change based on, again, this is average sized adult. There's a wide range in the size of people, and so this is a rough estimate. During pregnancy, females can increase up to about another liter to a liter and a half of blood. This is just showing you a picture. It's a pretty common picture here. You can see if you take a blood sample and you centrifuge it, spin it down, it separates into these 3 layers. The plasma is lightest, so it's at the top. You have this thin buffy coat in the middle. That's your white blood cells and platelets. So white blood cells, platelets, and red blood cells all together are the formed elements, but then they separate out when they're centrifuged into that buffy coat, and then red blood cells, the heaviest at the bottom. So looking at the formed elements, we're going to start with the erythrocytes, your red blood cells, your RBCs, all the same kind of thing. Just the general shape, they're these biconcave discs, so they're not round balls, they're not flat spheres. They have this certain shape that allows them to stack on top of each other, fit really closely into places, and as this note adds, it helps to increase surface area and puts the oxygen in close proximity to hemoglobin for it to bind, because we'll see those things are necessary. Red blood cells don't have a nucleus. They get rid of their nuclei. They really don't have many things in their cell except for hemoglobin. You can see a red blood cell is 1/3 hemoglobin. That's a lot. 30% of its contents are hemoglobin. And hemoglobin is what's necessary for transporting oxygen and a little bit of carbon dioxide. Name hemoglobin. So globin is a protein. So we'll see that this is like an iron pigment molecule that's kind of combined the hemo portion, and then globin is the protein portion. And so this thing kind of comes together, and the oxygen is going to bind to the iron within the hemoglobin molecule, and that's what allows it to transport through red blood cells. So when oxygen does combine with hemoglobin, it forms oxyhemoglobin, right? So oxygen, oxyhemoglobin. And think about when you incorporate, if you ever bake or anything, when you incorporate air into something, it kind of makes this lighter color, especially if you're baking like cookies or brownies or whatever it may be. You kind of whip the sugar and eggs and everything, and it gets that lighter, paler yellow color. And so by incorporating oxygen into hemoglobin, it gives them this bright red color. That heme pigment gets shifted. When oxygen gets released at tissues, then it's deoxyhemoglobin, and it's darker in color. It's not blue, it's more like a kind of dark red, purplish kind of color. It only looks blue to us because of how light passes through the skin, and so it appears to be blue, but really the deoxygenated blood in our veins is not blue. It is just like a darker red. Within red blood cells, they produce ATP through glycolysis, so that is an anaerobic state, that is an anaerobic process. So they are not using the oxygen that they're transporting to create energy for themselves. They are using a less effective method, but again, red blood cells don't really need a lot of energy. They don't have any nucleus, they can't make any proteins, there's really not a whole lot going on in there except for transporting oxygen and some carbon dioxide, maybe moving some things across their cell membranes. There's that structure, that biconcave disc that you can see, so 1, 2, biconcave, and this allows them to stack on top of each other. Here's the hemoglobin molecule, so you can see these kind of gold globin chains, and then the purple ones too. And then within them are these heme groups, these kind of red pigmented heme groups. And if you look, it's this kind of these circular carbon nitrogen containing rings. At the very center is this iron molecule, and so this is what the oxygen is actually binding to. The oxygen is binding to the iron molecule, and just like in your yard, if you leave something that is iron out in the elements for long enough, it's going to rust. And so that's what happens to this. It starts to oxidize. That iron molecule, by binding multiple oxygens over and over again, it begins to oxidize and break down, and so it has to be recycled. So these hemoglobin molecules, we'll see how they go about being recycled. Most red blood cell counts, it's 4.7 to 6.1 million microliters for adult males. That's microliters. That's what that little weird U stands for. And then for females, it's 4.2 to 5.4 million for adult females, so proportionally relative. The number of red blood cells determines your oxygen carrying capacity, and this is affecting your general health. The best way that I like to kind of describe this is thinking about athletes that get in trouble for blood doping, if you've ever heard of that, or when athletes go and train at higher altitude places. When you go up, to the mountains, especially, the big mountains like Colorado or Peru, where they have, much, much bigger mountains than we do over here, at least. Your body is naturally going to produce more red blood cells because there's less oxygen available to you at that higher altitude. And so athletes will sometimes go and train at higher altitudes to force their bodies to make more red blood cells. That then gives them a higher oxygen carrying capacity and more energy availability, because energy production is determined by oxygen availability for the most part. And so when your body is able to have more red blood cells, whether you do high altitude training or you do blood doping with something like EPO, which is a substitute hormone that induces red blood cell production into your body. Some athletes, some people will, I keep saying athletes, but lots of people do it, unfortunately, will just directly inject red blood cells into their blood. Rather than trying to make their own body make it, they'll just use red blood cells from a donor and use that. So lots of different ways that it can happen, and it is very dangerous. It is a very, very high risk procedure. That's one of the reasons it's not just regulated because it's fair to make sure that everybody's getting the same, you during competitions, that's what I'm thinking of, is you want to always make sure that people are being safe. It's not just about making sure that it's fair to the other participants, but it can be very dangerous to have artificially increased red blood cells. Makes your blood very viscous and thick. And so red blood cells counts are measured for diagnostic purposes and to trace the course of diseases. That red blood cell erythrocytes, that production is called erythropoiesis. Go ahead and learn this root, poiesis, P-O-I-E-S-I-S. We'll see it a lot this lecture. So erythropoiesis is the production of red blood cells. During the embryonic and fetal phases, it's produced in the yolk sac, the liver, and spleen once those start to develop. But postpartum, after birth, it's in the red bone marrow primarily. And hematopoiesis is what we call that, because in hematopoiesis, you're producing all your blood cells, all your formed elements. Red blood cells come from hematopoietic stem cells. We call them hemocytoblasts. And so we'll see that these stem cells have this ability to be able to differentiate not just the red blood cells, but also some of our white blood cells. The average lifespan for a red blood cell is about 120 days, so about 3 months. So every 3 months or so, you have all brand new red blood cells circulating through your body. Total number of red blood cells tends to stay relatively constant. It's a negative feedback mechanism, so that, again, makes sure that everything stays at a baseline level. The hormone erythropoietin is what monitors that, or what regulates that. That is determined by oxygen levels that are detected by chemoreceptors in your kidneys and liver. It's done in the kidneys and liver because you would think maybe the heart or maybe the lungs, but your kidneys and liver are circulating through a lot more blood than most of your other organs. And your kidneys in particular use about 25% of your body's available oxygen at a time. And so they're very much monitoring that level. If you have excess increase in red blood cells, like I was talking about on the previous slide, we call that polycythemia. Polycythemia. Emia always refers to blood. So polycy, increased poly, that's a lot, cyth cells, blood, increased blood cells, which we typically take to mean red blood cells, and so the most common formed element. That causes viscous, that's that thick, slow-moving blood, and ultimately oxygen deficiency because of that slow-moving process. So it's, again, very dangerous. Here is this this depiction of hematopoiesis, so production of blood cells. You start with this hemocytoblast, this stem cell, and stem cells have this ability to be able to differentiate into any kind of cell. And this one in particular is going to be a myeloid or a lymphoid stem cell. So myeloid stem cells then become your erythroblast, which become erythrocytes, megakaryoblast, which become platelets, myeloblasts, which are your white blood cells, 3 types of white blood cells. We'll see these are called granulocytes. And then monoblasts, which become monocytes. Again, another white blood cell. So this one myeloid stem cell line can produce 4 different types of other cells, which then themselves produce different kinds of cells. So this is just based off of hormonal factors, chemical factors that then differentiate what types of cells need to be produced. The lymphoid stem cell line produces lymphoblasts, which produce lymphocytes. And then I think in the immune chapter, in a couple of chapters, we talk about what happens to these lymphocytes. They become your B and T cells. So we'll see that more. But this is your red blood cell hematopoiesis, or I'm sorry, not just your red blood cell, but this is your hematopoiesis process in bone marrow. So all of this is happening in bone marrow. Here's just a little graph showing how that erythropoietin negative feedback cycle works. Low blood oxygen is detected by the liver and kidneys. They secrete erythropoietin into the bloodstream, which is transported to the bone marrow. That erythropoietin is going to cause an increased number of red blood cells. It'll use that hormone. It'll use some other nutrients from the blood to increase the number of cells, increasing the oxygen carrying capacity, and that has that negative feedback loop on this erythropoietin production. Other factors for red blood cell production, because it's not just the erythropoietin, vitamins B12 and folic acid for DNA synthesis is needed in general. So especially in this type of tissue, you have to have those kind of proper nutrients. Iron is necessary for hemoglobin synthesis. Most of that comes from recycling old red blood cells, not necessarily new iron uptake, but having that constant or consistent level of availability in the diet is still crucial for making sure that hemoglobin levels stay high. Deficiency in red blood cells or the quantity of hemoglobin can cause anemia. This is that reduction in oxygen carrying capacity of the blood. I think a lot of times when people hear anemia, they immediately go to iron, and that can be a cause if you don't have enough iron. As we were just saying, you don't have hemoglobin production, and then yes, that does impact your oxygen carrying capacity. But there are other factors and many different types. I've seen a couple of slides, many different types of anemia. But real quick, just this little vitamins B12 and folic acid are necessary. Same thing with iron. This is why it's possible to live off of Mountain Dew and donuts. I did that for years, and it was what I needed to do to get by. But over time, your body needs some extra nutrients. Like you can get by just on the calories, but at a certain point, your body is going to work much more optimally when it has these other nutrients available to it to be able to really meet its full needs. So that's just that difference between kind of just surviving off of calories and actually providing some good nutrients for your body. Do both. You need both. Here again is the different types of anemia based on either decreased red blood cells, decreased hemoglobin concentration, or an abnormal hemoglobin that can't bind oxygen completely. This one, the abnormal hemoglobin, this can result in sickle cell anemia, which again is limiting a genetic condition that can limit the number of hemoglobin molecules that a red blood cell can have and how many oxygens can be bound there. It causes this kind of half moon shape, and that actually also causes the red blood cell to be sharp, and it can cut capillaries as it travels through capillary beds. So sickle cell anemia can be dangerous for multiple reasons. What is kind of interesting though is that they've found that people who tend to have sickle cell anemia also tend to be resistant to malaria. And there seems to be some kind of genetic correlation that when you do have sickle cell anemia, it also somehow confers some resistance into malaria as well. And so it's very interesting how those things just sometimes go together. All right, so as we said, red blood cells only last for about 120 days, and then they're recycled. Things get reused, some things get excreted. So the breakdown of red blood cells, as they age, they start to become fragile. They're being pushed around a lot. They're being transported through the body. Think about, blood pressure can be intense. So they get damaged. As they get damaged, macrophages in the liver and in the spleen start to phagocytize those blood cells, those red blood cells, and break them down. The hemoglobin from the decomposed cells is broken down into both its heme and globin portions. Remember, the globin is a protein made of amino acids. The heme is both iron and pigment. It's this pigmented color. And so these two, the hemoglobin gets broken down into both of these things. Everything else, gets kind of eaten up by the macrophages. And then heme is broken down into its components, the iron and the pigment biliverdin. Biliverdin is then converted into bilirubin, and those things are excreted in bile. Sometimes it gets sent to the kidneys to be urinated out. So hemoglobin, red blood cell, think about kind of the shell of the red blood cell. That all gets eaten and broken down. The hemoglobin that's carrying oxygen, it gets broken into its heme and globin portions. The heme gets then broken down into iron and biliverdin, and then biliverdin has two routes, as bilirubin through bile, or it can be excreted through the kidneys. That globin protein, like I said, it's going to be broken down into its amino acid forms and then reused as it's able to be. And most iron, if there is excess, gets to be stored in the liver, but it can recycle back to your bone marrow for hematopoiesis there. So that's why, a lot of times people talk about how good liver is for you because it's got, a lot of iron. It's got a lot of iron, and it's because liver is storing iron. That's its job, one of its jobs. Liver stays busy. Here's just a breakdown of the red blood cell cycle. I'm not going to read through this, but this is definitely a good image to look at to make sure you can connect how this, especially these steps can be confusing when we start talking about the hemoglobin breakdown and what's going to happen to biliverdin. So just making sure that you feel comfortable with that. Okay, moving into white blood cells. So white blood cells are also called your leukocytes, leuko meaning white. This helps to defend against disease, against infections and pathogens. They're formed, same thing, from those stem cells, those hemocytoblasts in red bone marrow. White blood cells, what's different about them is that they actually can leave the bloodstream to fight infections. We call it diapedesis, dia meaning through and pede meaning kind of walking or stepping through. So they're stepping through the capillaries and actually going into the tissue, into cells, to be able to fight infections as they need. Outside of circulation, meaning outside of the blood vessels, white blood cells they use this amoeboid motion, so they have little stretching themselves out towards what they want and kind of pulling and dragging themselves along. That's called positive chemotaxis. Your infected cells, once they recognize that they're infected, or once they recognize their neighbors are infected, they start to send out these chemical signals, things like interleukins and colony stimulating factors, your CSFs. They start to send out infected cells, and neighboring cells will send out these chemical signals, and that's what attracts white blood cells into them. So there isn't as quite of a random exit of white blood cells as there would be for certain hormones, or for, like, when I was talking about ibuprofen last week, how it just kind of exits the body, and a lot of over-the-counter drugs and most drugs, it's going to exit the body. White blood cells, they are actually responding to these chemical factors, this chemotaxis. There are 5 types of white blood cells circulating through blood based on their size, granulation, shape of the nucleus, staining characteristics. All of those things are going to physically distinguish them from each other, and then we'll see their functional differences too. So this is just a general cell, white leukocyte. You can see it's traveling through this capillary, and capillary beds are right near the tissue samples. And so you can see this white blood cell squeezing through these little thin gaps in the capillary bed, one cell at a time, kind of creeping through to just monitor tissues and treat any infections that it finds. So let's break down the different types of white blood cells. There's 2 general categories, granulocytes and agranulocytes. The granulocytes are called that because they have this granular cytoplasm. It looks like they have little grains of sand or something in their cytoplasm of these white blood cells. Short lifespan, 12 hours or so, but they can still be produced a lot. You can still, relative for white blood cells, you can still produce a lot of them, even if they are in a short amount of time. And it kind of makes sense that they, don't last too long. These things are fighting infections. If we talk about red blood cells being kind of beat up and pushed around a lot, white blood cells certainly are getting that. Neutrophils, these are the most common, most abundant of your white blood cells. Purple staining cytoplasm, very lobular nuclei. So we'll see kind of how they have this kind of lobular appearance. I'm not sure how else to describe that, so you'll have to see that. And their main function is phagocytosis. They are strong phagocytes. They're going to engulf pathogens, break them down, and recycle those materials. So neutrophils are your most abundant. Eosinophils, these are for parasite infections typically. They also do have an effect on inflammation, but a lot of times when you see them, it's for some type of parasitic infection. I'm thinking, like worms and uh, those kind of different bugs. They have coarse granules. They stain this deep red color. It's very pretty. This deep red. 1 to 3% of circulating leukocytes. I'll teach you a nice, fun little mnemonic. I'll actually go ahead and tease you with it. It's never let monkeys eat bananas. So never let monkeys eat bananas. NLMEB. Neutrophils are your most abundant, and then eosinophils are the second least, and then basophils are your least abundant of the leukocytes. They're the bananas. They're at the very end. So they have few granules. They tend to stain fairly kind of bluish color, and their main function is heparin and histamine production. These are both, heparin is an anticoagulant, so it allows blood to flow. Oh, no, sorry, that's the, yes, yeah, that is correct. Heparin allows blood to flow. It's an anticoagulant, and histamine is used for inflammation. It helps to increase inflammation. That's why you might take an antihistamine, something like Benadryl, an antihistamine, because that helps to stop your nose from running, your eyes from watering, because histamines are there as an allergic reaction to help increase fluid movement, so that way you can get any kind of other nutrients, other leukocytes, other healing components that might be needed to an area. And so when you're taking antihistamines, you're kind of counteracting this basophil's job. So that's why we always say, if it's, unless it's really, really disruptive for you, try to allow your body to go through its normal processes, unless it starts to become excessive, right? So this is your neutrophil, just looking at a picture of it. You can see, again, that lobular nucleus. Here's a bit. There's a little thin line connecting this one to that one, those purple colors. This is a eosinophil. Like I said, this one's for parasites, that red staining cytosol or cytoplasm granules. And this is your basophil. So it doesn't quite look as blue. It almost kind of looks a little bit purplish, but I don't think you would confuse this one considering how dark this, there's the, you know, I guess this is bluish here, but you wouldn't confuse, let me go back, that, which is the other kind of purple staining neutrophil with its lobular nuclei. You wouldn't really confuse that with this. Those two cells look completely different. So basophils are your heparin histamine cells. The other type of white blood cells, so we did the first three, the last two are their agranulocytes. So they don't have a granular appearance to their cytoplasm. They tend to have a longer lifespan. These are your monocytes and your lymphocytes. Monocytes, they're the largest of the cells, and so they're the third most abundant. Lymphocytes are the second most abundant. Monocytes, they have a kind of kidney bean shaped nuclei that you're going to see, and they tend to go into tissues and turn into macrophages. So phagocytosis and macrophages, macrophages are a type of phagocytes, let's say that. And so depending on where they might be, where in the tissues in the lungs or the skin, the brain, they're going to have sometimes different names for those specific locations. But monocytes, macrophages, phagocytosis. Lymphocytes, we're going to see they turn into B cells, T cells, and NK cells, which are natural killer cells. Very kind of ominous name to them there. They're your second most abundant of the white blood cells. They can live for years, some of them. They are the smallest of all the white blood cells, though. They have a large round nucleus. And as I said, they are responsible for your immune system. So they, well, all of these are part of the immune system, but the lymphocytes are for your adaptive immune system, which we'll learn about in chapter 14, I believe. And so they're used for actually attacking specific pathogens. They have their own pathways. It's not phagocytosis. They use different techniques, B cells and T cells do, to be able to destroy pathogens. This is a monocyte. Like I said, it kind of looks like a kidney bean and its nuclei. And then this dark lymphocyte, you can see it's mostly filled with this nucleus. So white blood cell counts. We talked about hematocrit, that's your total blood cell count, but you can get specific to the formed elements. And so a white blood cell count, WBCC, or just a WBC, typical is 3,500 to 10,500 blood cells per microliter. That's the average, so it's a pretty large range, just depending on the people and A lot of times there's just small little infections that we're fighting, things that we don't really even notice, that we're not even aware of, we're not having symptoms of, because our body's capable of handling it. It's just doing that job of handling it. Little update. Right before COVID hit, Stanford had released a study that showed that the human body temperature had actually dropped a full degree, because the temperature that we were all most used to, the 98.6, that was from 100 years ago. So 100 years ago, in the late 19 or early 1900s, 1915 to 1925, uh, our medicine still was not very good. We still really didn't fully understand how dental health care was important, eye care, proper nutrition was important in maintaining a healthy body. And so a lot of people 100 years ago were just running fevers all the time because their body was constantly fighting infections. And so as medicine has improved, now our body temperature has dropped a full degree because we just are no longer fighting as many infections. So if you ever noticed while you were taking your temperature over COVID, it's like, wow, my temperature is usually like below 98.6. I feel like I'm running cold. Like, why is mine 97? Mine would sometimes get down to 96. Different thermometers was always kind of like a 96.9, 97.2. And it's because, yeah, you're not just running as many minor little infections that your body is fighting off. So that's why it's a range of such a large range. Those numbers can change. They can increase or decrease under abnormal conditions. Leukocytosis, that means an increase in your leukocytes. Things like acute infections, smoking, leukemia, they can cause an increase in your white blood cell count. Leukopenia, that's a decrease in your white blood cell count. Some viral infections, including AIDS, chickenpox, measles, influenza, things like that, they can actually start to decrease the white blood cell count. That's what AIDS is autoimmune deficiency syndrome. And so people tend to have the condition. If someone dies, typically they have the condition of AIDS, meaning that their white blood cell count is very, very low. That then makes them susceptible to something. And so most AIDS patients die of something like pneumonia. They die of something else, but it's related to this condition because their white blood cell counts are so low. And if they didn't have those low blood cell counts, they could probably fight off a normal infection. But thankfully, through medications, HIV and AIDS can be very well managed now. It's a completely different disease than it was even 10 years ago. And so it's really wonderful, the things they've been able to make and the strides they've made with treating that disease. Your differential blood cell count. So this actually, differential means that they're separating out the different white blood cells. So this 3,500 to 10,500, that's just for all 5 types of white blood cells. Generally, it should be in that area. Let's say you are fighting an infection and your doctor wants to look at why, they could order a differential to be able to actually see. Someone will go in lab and they can actually use a blood sample and they'll see, okay, are we mostly still seeing neutrophils? Are there a lot of eosinophils? Oh, we're mostly seeing eosinophils. Oh, this person probably has a parasitic infection. Let's treat it that way. And so that's how they'll kind of use differentials to be able to help make a diagnosis. Okay, platelets then. Moving into platelets. Platelets, also called thrombocytes, they are not actual cells. They're fragments of a cell. They're fragments of these megakaryocytes. Megakaryocytes are those large cells from the bone marrow, and what they do is they stick out their projections into the blood vessels, and then just as the pressure of fluid and things moving past the little projections, they break off little fragments of their own cell membrane, and those become platelets. Platelets, they don't have a nucleus. Again, they're just these little fragments, much, much smaller than red blood cells. They kind of move by that same amoeboid movement, kind of slinking across certain areas. Megakaryocytes and those platelets develop from that hematopoietic stem cells under the presence of thrombopoietin. So rather than erythropoietin, they use thrombopoietin. And when a myeloid cell is exposed to this thrombopoietin, it'll divert it down that pathway. Platelets help repair damaged blood vessels. What they do is they end up sticking to the broken edges. So once a blood vessel gets broken, let's say there's a cut or some kind of injury and it's leaking out, it's got an opening to it, platelets will go and they'll stick to each other, and they stick to the blood, to the damaged blood vessel wall, and create a clot. And that stoppage of bleeding, that clot stops bleeding, and that's called hemostasis. So hemostasis, that's a important word to learn, important word to remember. Hemostasis is the stopping of bleeding. Typical blood platelet count is 150,000 to 400,000 platelets per microliter of blood. Less than that, and then you might have a bleeding disorder. Your body might not be able to clot blood as it normally should. More than that, and then you might have a different type of clotting disorder where your blood is clotting too much and it's actually throwing off clots, meaning your blood is forming clots, and then those are traveling through your blood vessels and can be very, very dangerous. Because platelets don't have any kind of nucleus and they're just these cell fragments, they only live about 10 days. They're constantly being reproduced. Thrombocytosis, high platelet count, it can be infection, genetic defects can do that. Thrombocytopenia, this is a low platelet count, so penia is low. So when you see that, that means that there's a decrease in something. Thrombocytopenia, that low platelet count, genetic conditions, leukemia, radiation, some medications can cause it, autoimmune diseases can do it, and again, that increases the risk of internal bleeding. So even just some minor damage to a blood vessel, what to someone who has a normal platelet count might just be, a small bruise or something like that. For someone who has thrombocytopenia, a small bruise can turn into some severe internal bleeding. You just bump into a desk, or you think about how many times you just look down and you're like, oh, I have a bruise on my arm. Well, for people with thrombocytopenia, that can be pretty dangerous. This is just a chart going through the different components of blood, so you should definitely understand and know the components, descriptions, function, and then relative numbers present. If you can learn those numbers, good for you, good on you. I would definitely learn the different rankings of the white blood cells. So remember, never let monkeys eat bananas. Never let monkeys eat bananas. Neutrophils, lymphocytes, monocytes, eosinophils, basophils. That would be helpful. All right, so those were the formed elements. Let's look at plasma now and its role in blood. So plasma is that clear straw-colored liquid of the blood. We call it that because it's kind of got this yellowish tint to it, this yellowish color to it. Cells and platelets suspended in the plasma floating through it. It's mostly water, 92% water, but it has a variety of other things in there, mixed in there. Its main functions are to transport nutrients and gases, carbon dioxide to the lungs for exhalation, oxygen to the tissues for energy production. Same thing, it does a fluid and electrolyte balance, so making sure that all of those levels are correct, and maintain an optimal pH, that's 7.35 to 4.5. Most abundant dissolved substances are in the plasma, so not so much in the extracellular or in the intracellular or interstitial fluid. It's mostly in the plasma. These things are not used for energy production, though. The plasma does not have to produce these proteins are not used for energy. They remain in the blood and they remain in the interstitial fluids. And the three groups of those plasma proteins, your albumins, your globulins, and your fibrinogen. We'll see that albumins are there mostly for maintaining fluid balance, globulins for transporting things, and then fibrinogen is really important for blood clotting. So again, here you go. Your albumins, most abundant. They maintain colloid osmotic pressure. Colloid is a description of a kind of fluid. So if you think about, like you can have water, which is really thin. You can have soup. Like if you were making a soup, that would be thicker than water. And then if I said, well, what about a stew? Then that maybe kind of thinks of something even thicker. And so it's the same kind of thing. A colloid is just like a stew. It's like a thickened liquid. And that thickened liquid helps to maintain blood pressure, essentially. Blood pressure and fluid balance. We'll look at this more in the next chapter, but that's what the albumins are doing. The globulins, like I said, the different kinds, they're transporting stuff, working as antibodies, and then fibrinogen for blood coagulation, blood stoppage. So the most important blood gases, those things that are transported through the plasma, those are oxygen and carbon dioxide. And then the nutrients, we talk about nutrients because that's a nice vague word to be able to throw around. We're talking about things like amino acids, simple sugars, nucleotides, and lipids to be able to make macro molecules. Vitamins and minerals for things like DNA synthesis and hemoglobin production, as we've seen already in this lecture. All things like that get absorbed from the digestive tract. Glucose that's traveling through the plasma, it can be used for energy or transported to the liver for conversion to glycogen or fat. So using it for energy, meaning transporting it directly to cells to let them just use that glucose. Or if there's enough excess, then it gets converted to something else. Amino acids for proteins, and sometimes for energy if needed. And then lipids, they're not soluble in water, so they actually bind to proteins to help them get transported through the bloodstream. So that's something always important to remember. We talked about that with hormone transport. Lipid hormones are going to be bound to carrier proteins, those globins. There's also non-protein nitrogenous substances, NPNs, non-protein nitrogenous substances. So most proteins are your main source of your nitrogen compounds, but there are some other NPNs that, or some other nitrogen sources that are not proteins. Things like amino acids, which are protein building blocks. So you can still have free amino acids in your body that haven't been converted into proteins yet. Urea and uric acid that can be made from your cells and your kidneys as well. And then creatine and creatinine, which creatinine is a breakdown of the creatine molecule. Urea and uric acid, again, byproducts of protein and nucleic acid catabolism, nucleic acids for your DNA. Right, and amino acids from breakdown of proteins. So all of these sources, as things are being recycled, get transported and travel through your blood, either to the kidneys, to the liver for excretion. Or recycling, right? Electrolytes in the plasma, so we talk about electrolytes a lot too and how important they are. An electrolyte is something that is going to break apart once it gets dissolved. So things like salt, sodium chloride, you put it into water, it doesn't just dissolve, it also actually separates into its positive sodium ion and its negative chloride ion. And so sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, sulfate ions, these are all your important electrolytes, with the two most important being sodium and potassium. These levels are very tightly regulated by several different mechanisms in the body, and those, along with things like calcium, are crucial for nervous system function and muscle function. And then you probably know people that take magnesium to help themselves sleep, take other things to help themselves, their bodies regulate and supplement. You should always consult your physician, make sure that you're doing what's right. And then some just work to maintain that osmotic pressure and pH of the plasma, as we've been talking about. Osmotic pressure, this is blood pressure. That's pretty much what that is. So here's this picture again of blood. Maybe it makes a little bit more sense now. When you take blood and break it into its formed elements in plasma, hopefully you at least understand these different titles and these categories, what's being contained within them, and what their basic functions are in the body. So let's now get into hemostasis a little bit, the stopping of bleeding. So let's say, you are cooking and you accidentally cut your finger, slice your hand or something, and you start to bleed. There's standard 3 processes that are going to happen, whether it's an external wound or an internal wound. You have first your vascular spasm, your vasospasm, the platelet plug formation, and blood coagulation. And typically, we can kind of think about them as 1, 2, 3, but there might be some overlap that's happening in these, just as different, depending on how large the wound is, and if different parts of the body are responding differently. So let's get into these 3 processes for stopping blood loss. The first is this vascular spasm. So this is essentially after a small blood vessel, you cut it after you cut a wound in a blood vessel. It's actually going to cause that smooth muscle to contract, smooth muscle walls to contract. So vascular spasm, a spasm in your calf muscle or a cramp in your calf muscle, same kind of thing here, except this is happening in a blood vessel. That reflex, it only lasts for a few minutes, but it causes a strong restriction in blood flow that can last for 30 minutes. And what's nice is that that's about how long it takes for the other 2 steps to start to form, start to initiate. And so right at that, this is a local response, right? So right at that place where the blood vessel was damaged, those smooth muscles are going to start to constrict, and it's really going to slow down blood flow in that area. That's why it's also helpful to add pressure to a wound, because adding pressure to an open bleeding wound is going to help kind of make sure that the blood is literally just staying in. You're literally just holding it in there, and you have to usually put a lot of pressure to help with that, but it can again, assist in this vascular spasm phase until these other 2 phases start to initiate. Even if you just have, a small cut on your hand or something, the instinct, or, at least my instinct, is not to just like hold it out and wave it about. I'm going to grab a paper towel, I'm going to grab something, I'm going to put it over top of it, and I'm going to push. I'm going to hold to try to keep that, to assist this vascular spasm phase. Platelets also release serotonin that also helps to constrict smooth muscle. That vasoconstriction decreases blood loss at the site of injury. It does not affect blood flow in general to the body spot. That is why you're still able to have these other two phases happening, because these phases do require some movement of blood to the area. So the platelet plug formation is the second phase. Typically, when you have an intact blood vessel wall, it's very smooth and it actually produces a certain molecule that kind of keeps it slick, kind of almost like a wax material that helps to make sure things slide along the inside of the blood vessel wall. But once that blood vessel wall is broken open, it actually starts to that chemical, that molecule it was producing, that this smooth endothelial linings of blood vessels, this molecule that these endothelial cells are producing, they actually stop producing it. So that then helps the platelets to stick a little more to the inside of the cell. And then also, because once the cell is broken open, it starts to expose connective tissue, the collagen fibers of connective tissue. And so that really makes the platelets stick. So kind of think, about, and we'll see in a picture here in a moment, a blood vessel broken open, and as it's exposed, it now has all these sticky collagen fibers that were internal now being exposed to the external cellular environment. And then the platelets will stick to that. So they adhere to those exposed edges, and they start to make they kind of create these spiny projections off their cells that protrude from their membranes, and they all stick together. And so this forms that platelet plug at the site of a break in a blood vessel. It's most effective on a small vessel because platelets are pretty small themselves, and if blood pressure is high enough in a certain area, it can kind of just cause that clot. The platelet plug can never really fully form, and so at just small vessels, it's most effective. Here you can see the platelet plug formation, this break in the blood vessel wall, and then the leaking of blood out of that escaping space. But these little ragged fibers right here, those are those exposed collagen fibers. And that's what the platelets start to stick to. They start to stick to each other, and they form this plug. And then you can get into the blood coagulation phase. So this is the most effective means of hemostasis. It is a chemical reaction. So whereas the platelet plug was mostly a physical reaction, coagulation is a chemical one that actually forms a full blood clot. So you have a platelet plug, temporary, and then a blood clot can be the long-term solution. It's very complex. It uses a lot of different clotting factors that you don't have to really learn at this point, but there are a few that you need to get through. So the first step in the clot formation is that the tissues that were damaged, the cell walls that were damaged, they start to release tissue thromboplastin, and then that creates prothrombin activator. That prothrombin gets converted into thrombin, and then thrombin converts that fibrinogen into fibrin. And that's really what allows for a blood clot formation. So it's step, 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 step. We'll see in the slide or two a nice little graph image of that. But just understand that it's these different hormonal factors or chemical factors kind of activating one after the other in the presence of calcium that allows it to form this fibrin network. So those threads, those fibrin threads, think fibrin, fiber, threads, right? So fibrin threads form that network that help to trap cells and platelets, and they form an actual clot. And the blood plasma starts to change. They have all these different factors and things active now. And so it goes from just a liquid form to more of a gel form, so a little bit thicker. So here's a nice little depiction of this, all three mechanisms of hemostasis. Tissue damage first, some kind of cut or a break of the blood vessel. There's the vascular spasm, the platelet plug formation, and then lastly the clotting mechanism. Clotting mechanism has that prothrombin activator. Prothrombin gets turned into thrombin, fibrinogen into fibrin, and then that forms that blood clot formation. Here's just a picture of that. You can see these red blood cells kind of covered by this fibrin mesh, sticking them all together. So these are not platelets, these are red blood cells. So after the clot formation, a clear yellow fluid remains in the blood. That's called the serum. Serum is plasma minus the clotting factors. So you might have heard of serum before. Sometimes that's what they'll do is when you go to give blood or have blood drawn, they take a serum sample. And that means that they take out, anytime you have blood drawn, you have your whole blood drawn, right? They pull it all out and it's all the stuff. It's red blood cells, white blood cells, platelets, your formed elements, your plasma, everything. But then they can take it to that centrifuge and they can spin it down, they can separate out what they want, and then they can actually put stuff back. So sometimes while they do take a whole blood draw, they might only save your red blood cells and then give you back your plasma. They'll actually put your plasma back into your body for you, your white blood cells, all that stuff. So that way you don't have as big of an effect on your body system from donating blood. Same thing, they can take plasma and they can remove clotting proteins. They can remove certain factors in it and create just a serum to give to somebody. And so serum is what's left in these blood clots. And if you ever think about it, if you've ever had maybe a cut or a scab and it starts to leak and it has that kind of clear yellow color to it, that's your serum. That's your plasma without the clotting proteins. If it turns into pus, which has that whitish, thick viscousness to it, and maybe sometimes a small smell or odor, pus is actually serum plus neutrophils. So usually when you see pus, you think infection, and that's what you're taught to kind of think. And that's true because if you see pus, that means that neutrophils have been drawn to that area to fight some type of pathogen. It doesn't mean that you have an active infection. It might have just been, at that point, there were some bacteria trying to get in. The neutrophils took care of it. It's no big deal. But now those neutrophils are dead and they're just kind of hanging out in this blood clot area because they got stuck there. And so if it's nice and clear, it's just that serum. If it's a little pus colored, it's probably some neutrophils in there. Prothrombin activator is proportional to the amount of tissue damage. That's why blood coagulation is kind of the most effective form, because it will be able to match whatever level of tissue damage there is to a degree, right? This is a positive feedback system, and so while that can be, intense because it's, kind of really throwing you into these unstable conditions, because it's a temporary reaction, which is trying to minimize this blood loss, that ultimately turns you back into a homeostatic internal environment. And so that's the goal here. So blood clotting, yes, it's a positive feedback mechanism, but once it puts you back into minimizing your blood loss, it starts to be minimized itself. So once you have a clot formed and the blood has stopped and wound healing has begun, you eventually have to dissolve that clot. So clot dissolution, what happens, sorry, I got lost. Let me read this first thing. So circulating blood prevents clot formation in uninjured areas, transporting excess thrombin away from these areas. That's one of the nice things about having this constant circulating blood, and even while in a wounded area, there is still some blood circulation happening to be able to make sure that there's not an excess clotting that's going to be formed, or clotting forming in unnecessary areas. A clot, as a clot due to blood leakage heals, clot dissolution, this fibrinolysis occurs. During fibrinolysis, breaking up of the fibrin, plasminogen is converted to plasmin, and plasmin is what's going to digest those fibrin threads. Clot that forms abnormally in a vessel is called a thrombus. So sometimes you might hear of someone having a thrombus in their lungs or something like that. That's a blood clot that's somewhere where it's not supposed to be. And then that thing can travel. And if you have a traveling clot, it's called an embolus or an embolism. And so pulmonary embolisms, those are, again, something very dangerous in the lungs, a clot that can be traveling through the lung or blocking part of the lung. Coronary thrombosis, a clot in the vessel that supplies the heart. Infarction, something that blocks blood flow and kills tissues. Myocardial infarction, right? And atherosclerosis. Atherosclerosis is a hardening of the blood vessels, usually due to fatty deposits, plaque, cholesterol buildups. This is a picture of atherosclerotic vessel. So you can see this is kind of normal tissue. You got this muscular layer, this inside of the artery wall where the blood would be flowing, the lumen. And then you can see here how much smaller that lumen is, because you have this thick plaque layer that's been building up along the artery wall, and it's really decreased how much blood can flow through an area at a time. So it's decreasing the blood flow, but also these plaque deposits can start to flake off if they start to get like a little dislodged, and that can cause a blood clot. So now let's look at blood groups and transfusions. So this is kind of one of those cool things about human science, is that we're able to transfuse blood from one person to another and truly give them life. So it was a process. Which is very scary to think about. This learning process that not there are different types of bloods. We can't just do one for the other. In dogs, I believe there are some different blood types in dogs, but there are fewer blood types, and so it's easier to match with them. Not that you can accept dog blood, but I mean between dogs, and with other certain animals. In humans, though, they found out that really does make a big difference. The types of blood really have some incompatibilities for transfusions. So before a transfusion is given, the donor and recipient blood have two tests: ABO and RH status, and cross matching. So donor, that's the person that's giving their blood, and the recipient is the person that is receiving the blood, that needs the blood. So they both kind of go through this stuff. The ABO, this refers to the three different blood types: A, AB, B, or O. We'll look at them. And the RH factor. This is the positive or negative, so A negative, B positive, that kind of thing. That's what the RH factor stands for. Really, it's the rhesus factor is the full name. And if possible, those should be the same. You want your donor and recipient ABO and RH to match completely. We'll see that there are some exceptions, there's some wiggle room, but if they're able to, they match them exactly. The other test, that cross-matching, that's when they actually take a small sample of recipient serum with donor blood cells to check for agglutination. So let's look at that real quick. Recipient serum with donor red blood cells. They don't have to mix the whole blood. So the reason is because the antibodies that can cause agglutination, that can cause a reaction to donated cells, they exist in the plasma, in the serum. And so they'll just mix a little bit of whoever's supposed to be receiving this blood. They'll just take a little bit of the serum that has their antibodies in it, their proteins, right? Antibodies are proteins that has their antibodies in it. They'll mix that with the red blood cells of whoever's donating the blood. And if it clumps, then they know, oh no, this is not a good match. It's kind of a quick way to check before you actually do a full transfusion. So breaking down some of those words, antigens and antibodies. An antigen is a molecule that evokes an immune response. So a molecule that stimulates some type of response from the immune system. I like to think about antigen generates a response. So an antigen is something that is normal to your body. It is sitting on your blood cells or sitting on all of your cells, and it's an identifier. It's like how a ship would fly its flags and they fly their colors, so that other ships know, oh, are you safe? Are you dangerous? Are you pirates? Should we be worried? Blah blah blah. That's what an antigen is doing. It's a molecule that sits on the surface of cells, and it's a way to identify itself. Bacterial cells will have antigens on their surface, and your body can recognize those, and antibodies will bind to antigens. So antigens are on the cells, identifying them. Antibodies are proteins floating through the plasma that are going to bind to antigens that are foreign to a cell or that are dangerous to a cell or to a body. And they will cause that clumping if it's a red blood cell. So if it's an improper transfusion, they agglutinate, they stick together. The antibodies from your plasma, or from the recipient's plasma, not your plasma, the antibodies from the recipient's plasma will agglutinate and stick to the antigens on the red blood cells of the donor. That's why they do that cross-matching test. So when those transfusions occur, antibodies and the recipient's plasma agglutinate on the surface of the donor's red blood cell, and that is a transfusion reaction is what they call that. So transfusion reaction is that agglutination reaction. They can produce very serious effects. Anxiety and breathing problems are some of the least of them. They can go up into jaundice and kidney failure. So they can be really, really deadly. There are 32 different red blood cell antigens, so meaning 32 different markers on red blood cell surfaces, but not all of them are going to produce transfusion reactions. The only ones that really do that are the ABO and the RH groups. So ABO antigens, RH antigens sitting on the surface of red blood cells, they can trigger the recipient's antibodies to stimulate an immune response. So let's look at the different ABO blood groups. We call it ABO because those are the three different types. The three different types of antigens that can be on a blood cell. The thing is that O actually stands for nothing. O is really kind of a zero because there's no antigens on people that have O blood. So the blood groups are based on the presence or absence of A or B. antigen A or antigen B on the red blood cell membrane. So the surface of the red blood cell is going to have one of these, both of them, or none of them. Antigens A and B, they're carbohydrates that are attached to glycolipids, and blood types are inherited. So it's based on genetic factors from your parents. Type A blood has A antigens and anti-B antibodies. And that's how we kind of phrase this. So kind of go ahead and listen to that a couple of times, read it to yourself a couple of times. Type A blood has A antigens on the red blood cell and anti-B antibodies in the plasma. And this anti-B antibodies, it means that it has these antibodies, these proteins in the plasma, in the serum plasma, that are going to attack any B antigens that it sees. Sorry, that any B antigens that it sees, and made me laugh. Type B blood has B antigens on the red blood cell and anti-A antibodies in the plasma. So these anti-A antibodies will attack the A antigens on red blood cells, just like the anti-B antibodies would attack the B antigens on those cells. Type AB blood, that means that your red blood cells have both A and B antigens. And since they have both A and B antigens on their red blood cell, there are actually no antibodies against them in the plasma. If someone had AB blood and they had antibodies in their plasma, then they would have this kind of autoimmune disorder where their body is just constantly attacking their own blood. And so that would be very bad, very dangerous. Because this type AB blood, does not have any antibodies in the plasma, we call it the universal recipient, because basically they can, broadly speaking, they can receive any type of blood, A, B, or O, and because they don't have any antibodies, their body will be fine with it. It will not work, typically it does not function quite as well. There can be some other issues, but for the most part, they can be a universal recipient for any other type. The last one is the type O blood, and again, O has, standing for O for nothing. It has no A or B antigens on the surface of the red blood cells. However, this means that it does have both types of antibodies. So this one is the universal donor. Because O blood, and when you're donating blood, sure, they take out all of your blood, they take out your whole blood, they centrifuge it. When they go to actually do the transfusion, they're usually only giving the red blood cells. So even though type O blood has antibodies for A and B, all they're going to be donating is the red blood cell with no antigens. So type O blood is your universal donor. It can go to AB, B, or A and not cause a reaction. However, it can only receive O. Since it has both types of antibodies in the plasma, if someone with type O blood is receiving, they have to get type O blood. They can't get any other kind, or their body will attack it. Here's what that kind of looks like. So you have type A antigen with anti-B antibodies. Here's type B antigen with anti-A antibodies. Here's someone with type AB blood. You can see they have both antigens. And then type O blood, there's no antigens, but there's both antibodies. So for blood transfusions, certain combinations have to be avoided. Antibodies of one type react with antigens of the same, causing that agglutination. So anti-B antibodies with B antigens will agglutinate. Main concern is that agglutination, and again, there's just an example of it, what I was just saying. Here's what an agglutination reaction might look like, a transfusion reaction might look like. This is someone with A blood, so antigen A on the cells, that means it's type A blood with anti-B antibodies. Let's say that they are given a transfusion that contains anti-A antibodies. Well, then those anti-A are going to bind with those A antigens on the cell of the surface of the red blood cell, and then they cause this clumping reaction. You can see how they start to clump together. Right in there, that's kind of what's happening. Here is just based on strictly ABO blood typing, the type of blood, the antigens that they have, and the antibodies that they're going to contain. So you should understand this chart if this helps you to see it in this way, but you should understand kind of all this information, how type is going to determine, or rather, when you hear a type of blood, that should immediately help you understand what type of antigens, if they're present, are there, and then same thing with antibodies. So again, for universal donor and recipient, if possible, it's always best to use the same blood type. If not, some substitutions can be made. The universal recipient is that type AB blood. Since it's type AB, it means it has both antigens on the red blood cells but no antibodies. So they can receive transfusions from any of the 4 blood types. That universal donor, the type O blood, since it does not have any antigens on its cell surface, it can be donated, transfused to any of the other 4 blood types, since its red blood cells won't be attacked by any antibodies. There's nothing there to trigger it. It can kind of just act as this nice little under the radar carrying oxygen for the body, but not really understanding. The body doesn't really recognize it's there. Here is preferred and permissible types for blood transfusions. So blood type of the recipient. And when you're looking at transfusion stuff, always make sure you're being clear about the recipient and the donor. Because the blood type of the recipient, this is where you have to really make sure you have to think the recipient is what's going to have the antibodies in its plasma. The blood type of the donor, you're only really donating red blood cells. So it doesn't matter what type of antibodies the blood donor has. It's all about the antibodies the blood recipient has. So this is the preferred type, and then if preferred is unavailable, you can see the O, it can go to any of those other 3. No alternate types for O blood. That's sometimes people with O, especially O negative, that will see the Rh stuff. People with certain O blood types, they will go and donate blood regularly to have blood stored in case either they get hurt or blood banks. I have friends that have O blood, and they get called regularly from the blood banks asking to please come in and donate, so that way in case they can always have O blood on stock, because it does act as that universal donor. And for people that have type O blood, they always like to have a good stock available in case it's needed, because there is no alternative. So that Rh group, I mentioned it kind of very vaguely. The Rh group was named after the rhesus monkey. In humans, there are several Rh antigens. The most common is the antigen D. Don't ask me why it was named after a monkey. I don't know. But this is the positive and the negative that you sometimes hear. O negative, A positive, AB negative. It's that negative or positive factor. And really what it's saying is positive means that the antigen D is present. So it's Rh positive. And if that that D antigen is absent, then it's Rh negative. So we've got A antigens, B antigens, and now D antigens. And present, positive, absent, negative. No corresponding antibodies in the plasma, unless this is very kind of specific, so this might sound confusing. Wait until the next slide. So no corresponding antibodies in the plasma, whether you're positive or negative. So if you're born Rh positive or Rh negative, you still won't have any antibodies in there unless a person with Rh negative blood has physical contact with Rh positive blood. Then the Rh negative person will develop anti-Rh antibodies. Let's talk about how that happens. 2 main ways in which someone with an individual with an Rh negative blood contacts Rh positive, either through a transfusion, through a mismatch, or through pregnancy. So transfusion, one's pretty straightforward. You give somebody, and that could be something like you had a limited supply and weren't checking carefully enough, and so you just give somebody Rh negative, Rh positive blood. The more common one, I believe, is through pregnancy. So let's say that the mother is Rh negative. She has Rh negative blood, but her fetus is Rh positive. And so she can be exposed during that delivery. She can be exposed to that Rh positive blood. There can be some mixing that happens there. And if it's a big enough exposure, that Rh positive blood will mix enough to cause and stimulate the Rh negative mother to produce anti-Rh antibodies that could attack the blood of a future fetus that's also Rh positive. So, if you kind of think like big picture down the road, like it seems like it would be like, oh, that seems very rare. It's not that rare. It happens a lot where someone who is Rh negative is going to give birth to an Rh positive child. And so if that Rh positive child's blood gets mixed with the Rh negative from the mother, that mother is going to produce those antibodies. And let's say that mother gets pregnant again and it's another Rh positive child, because Rh positive is a dominant trait, negative is the recessive trait. And so if the mother has if the father rather of the second child is also Rh positive, and then that will cause that Rh positive child to then potentially have their blood attacked by the mother's blood. We call that hemolytic disease of the fetus. That's normally what I've heard it called. Erythroblastosis fetalis. I've not heard that before, but hemolytic disease of the fetus or newborn, that is kind of a more common name what you hear. They can be prevented. They give an injection called RhoGAM. So if the mother is Rh negative, at around 28 weeks of pregnancy, they give her a RhoGAM injection, and so that just prevents the mother's immune system from producing anti-Rh antibodies. It has to be done if she gets pregnant again. It's not a one-time thing. It has to be boosted, in a sense. This is what that would look like. So you have this Rh negative female with an Rh positive fetus. Fetus in here. And then their blood starts to mix. So this D antigen exposes the mother to it, and then she starts to create these anti-Rh antibodies. Because it's just a small amount of blood, like it looks like a lot of blood here, like half of her blood cells that we're seeing, but it's usually just going to be a small enough blood. Again, enough to cause this reaction, but not enough to cause, a full agglutination reaction. And let's be very honest, during pregnancy, during childbirth, a female's body is going through so much, some, lightness of breath and some anxiety, some, stress, like that's going to kind of a normal thing, I think, for a lot of childbirth practices. So there's not going to be necessarily a noticeable agglutination reaction in the mother, but again, you have these lingering antibodies, and then if she gets pregnant again with a Rh positive fetus, well, then her antibodies can transfuse and agglutinate in the fetal blood, and that can be fatal. So here is the full chart of preferred and permissible ABO and Rh blood types. So putting in the negatives and the positives. So blood type of the recipient, you can see negative has to give to negative. Positive would be good for it to give to positive. However, an A positive can also take from A negative. Same thing. B negative, best to give B negative. You can give O negative if you need to. B positive is the preferred, but it will accept B negative, O negative as well, universal donor, and O positive. It can actually take O positive because it's a B positive. Notice that B negative cannot take O positive. So truly, we said O is a universal donor, but truly, it's the O negative is the universal donor. And as we'll see with AB positive, that's your universal recipient. AB positive can receive AB negative, A positive, B positive, O positive, because it doesn't have antibodies for anything. Whereas O negative, because it does not have any antigens for A, B, or that Rh antigen, there are no alternatives for what it could be donated. You can only accept O negative. O positive, because it's positive, it doesn't have any, there's no risk for this blood to be producing anti-Rh antibodies, so O positive can accept from O negative as well. All right, I think that's the last slide. Yeah, so that last bit about blood transfusions can get confusing. There are some good videos on Connect, so make sure you're reviewing those videos, and then we'll try to do some activities in lab on that. All right, thank you so much.